Today, I will be illustrating two important experiments in the history of the genetic code. To preface, scientists already discovered that RNA sequences made up of a combination of 3 nitrogenous bases coded for specific amino acids. Their next step was to figure out which 3 letters or bases coded for which amino acids. 2 experiments were done to figure this out. First, an experiment was done by Nirenberg and Mattaei. In a test tube, they added poly-U homopolymer, cell-free extract, and added all 20 amino acids, with 19 of them unlabeled and 1 radioactively labeled. They did the experiment 20 times, and each time they had a different radioactively labeled amino acid. Translation took place, and the resulting protein, a string of amino acids, was checked by the filter for radioactivity. If the resulting protein was radioactive, then the labeled amino acid must be in the protein, and that means the codon UUU codes for that amino acid. They found that the radioactive label only appeared in the tube for phenylalanine, which means that UUU codes for that amino acid. They repeated this for a poly C and a poly A, and they were able to identify 3 out of 64 codons. In order to figure out the remaining 61 out of 64 codons, a second experiment was performed by Nirenberg and Mattaei. They combined short mRNAs with all the known codons on them and added them to a mixture of ribosomes and tRNAs attached to amino acids. They used a somewhat similar procedure where 1 amino acid was radioactive per each round of the experiment, and they did 20 rounds. They allowed binding to occur to see if the particular radioactive amino acid would bind. Using a filter, unbound amino acids would filter out, and if the ribosome bound to the tRNA and mRNA codon, they would be caught by the filter paper, and it would mean the trinucleotide codes for the radioactive amino acid. For example, when an mRNA with GUU was added, the tRNAs on the filter paper were bound to valine, meaning GUU codes for valine. Nirenberg and Mattaei identified the remaining 61 codons in this experiment. With the conclusion of both experiments, scientists were able to identify all 64 codons.